So now it is my pleasure to introduce Professor David Amor as our first speaker. Professor Amor is, you're welcome, well, come up, David. Professor Amor is trained in paediatrics and genetics at the Royal Children's Hospital and has a PhD in chromosome biology. Previously, he was a director of the Victorian Clinical Genetics Services, and he's been appointed to the Garley Chair in Developmental Medicine at the University of Melbourne in 2016. David's current research focuses on the genetics and intellectual disability and rare disease syndromes. And he also focuses on the translation of new genetic technologies into clinical practice. Please make David welcome.
Thank you very much, Linda. That introduction was a bit long, wasn't it? I'll trim it down for next time. So thank you very much to both of you and to Heather for asking me to speak. It's a great pleasure and honour to come here, even on a Saturday morning. And also, you've given me a bit more time, haven't you? Because I wasn't due to start for another 10 minutes. So, yeah, so that's fantastic, because I was looking at my talk this morning. I thought, gee, I'm going to struggle to get through this in 25 minutes. And Sue generously said I can have some of her time, but maybe I won't need it. And you might even get a bit of extra, Sue. Okay, so this is kind of the genetics 101 type talk. So it's probably not the most exciting talk that you're going to hear today, but it is important as a starting point, so you can maybe make sense of some of the stuff that will come after, and I'll try and make it as interesting as possible. Some of these slides, as you can see, I stole from the Melbourne Genomics Health Alliance, so I give them credit for the slides. In genetic counselling, we're often trying to talk to or explain to patients about exactly what genetics means and how we take very complicated science and put it in a patient-sensitive manner or an understandable manner. So when we think about genomes, we sometimes use this analogy of a person's genome is like a book, or sometimes it's even like a set of encyclopedias, and then we can break it down from there. So within the book, there might be individual chapters, which we can think of as chromosomes. And then with each chapter, we can think of individual paragraphs or sentences that are like genes. And then we get down to the spelling mistake in the gene, which is often the type of genetic change that ends up causing health problems. So how does this all come together? Well, within our bodies, we have cells, and the body is made up of billions and billions of cells. And in nearly every cell, there is a nucleus, red blood cells being the exception. And if we drill down into the nucleus of each cell, we have chromosomes, and there are 23 pairs of chromosomes usually in each cell. And it's within those chromosomes that the genes are contained. In addition, humans have a second genome, which is often forgotten about, which is the mitochondrial genome, and it has its own special form of inheritance that is being passed on only from the mother to the child. And there are a number of genetic disorders and syndromes that are inherited not from the nuclear genome that is in the chromosomes, but from the mitochondrial genome. But I'm not going to go into detail about those today, given the limits of time. So I quite like this picture because it kind of gives you an idea of the scale of when we go from a chromosome, which is this enormous unit down the bottom, and then inside the chromosome, DNA is wrapped around histone proteins, and it's arranged in a sort of secondary and tertiary conformation into the chromosome. But when we're talking in genetics, usually we're talking either about changes at the level of individual base pairs along the DNA, or we're talking about whole chromosomes. And in the olden days, we used to look at chromosomes down the microscope like this, and some of you may have seen a picture like this in your high school biology days. And so, you know, just a reminder that there are 46 chromosomes altogether in a human genome, made up of two sets of 23 chromosomes, and the chromosomes are numbered according to their size. So chromosome 1 is the biggest chromosome, and chromosome 21 and 22 are the smallest. And if we unravel each of these chromosomes along the lines of the figure on the left, you can you would then end up with either a couple of hundred genes in the case of chromosome 21, or several thousand genes if we're talking about the biggest chromosomes. So if we look just more closely at DNA, this is the DNA double helix. It's well recognized or recognizable, and the DNA is made up of individual nucleotides. So these are the letters of the genetic alphabet, so A's and T's and C's and G's. And in cell division, you end up with one copy of the DNA, or the two DNA strands separate, and then a new second strand of DNA is produced, and that's how cells divide and replicate. So let's talk a bit more about genes. So within our body, we have about 23,000 genes spread across the chromosomes, and they all do really important jobs, but they're not all doing the same job in the same cells at the same time. So along a particular chromosome, in this figure, this might be a particular gene which might do a certain role in development for a period of time and then get switched off. And then another gene might get switched on in a particular cell and do a different job. And each gene is a recipe to produce a particular protein. So one of the interesting things about the human genome is that there's a lot of DNA there that does not actually code for proteins or does not make up genes. So many of you are probably familiar with the term exome, which is basically the term used to describe the parts of the human genome which encode for proteins or parts that are genes. And we now recognise that the exome, or the protein encoding part of the genome, is only about 1 to 2%. And people are still working out what the rest of the genome, the other 98 to 99%, is doing. So it used to be thought that it was just junk or filling in the gaps, but it's now recognised that a lot of that material is has a role in fine-tuning the function of the other genes in the genome. So it is doing something important, or at least some of it is. Okay, so let's think about an individual gene, and this is a particular gene, which is human keratin. It's a gene which encodes for a keratin protein, which is important in hair and nails. And so this particular gene has 1,861 coding bases. So that's the number of letters in the gene, or the numbers of individual, the number of nucleotides in the gene. When we look at genes on the computer, often we look at it in this way, where it's all divided into 10 nucleotide blocks. That just makes it easier to read and work out where you are. But of course, all of those gaps are not actually real. So this particular gene, keratin, we can think of changes in that which might lead to normal variation between people. So it might affect something about your hair, and might also affect disease. So changes in genes can cause variation between different people, which is just part of, which is not specifically health related, but they can also cause human disease. So I'm going to talk a bit more about variation in genes, and this is what we tend to look for when we do genetic testing. And there's two main types of genetic variations. So the first is what we call single nucleotide variants, that is individual spelling mistakes in genes. And the second is chromosome variants. And when we think about the children in particular that we see in our clinic, most of them in whom we are able to make a diagnosis, we would find they fit into one of these two categories. So either they have a single nucleotide variant or a chromosome variant. So the single nucleotide variants, we start off thinking about the reference human genome sequence. So that's what we regard as normal, if you like. And here, for example, you see one letter has changed from a T to a G in a particular patient. Chromosome variants are a little bit different, so they can take different forms. So for example, we might see a translocation, an exchange of material between two chromosomes. We might see a deletion, little piece chopped out of a chromosome, or we might see an insertion, a bit of extra material put into a chromosome. So regardless of whether we're talking about single nucleotide variants or chromosome variants, variants can have different consequences. So a lot of them are neutral, that is, they don't have any effect on health. Rarely they can be advantageous, so they can result in improved health or be, so improved health, and sometimes they are deleterious, so that means they can cause disease. So the way we think of that in a medical context is some of them can be benign. So when we use the term benign, we tend to think of them as harmless, and then we use the term pathogenic to describe variants that cause disease. And sometimes we're not really sure, so there are also variants that we would say are of uncertain significance, and that means maybe they contribute to disease, maybe they don't. And that may be something that we can sort out in time. So maybe if we come back a few years down the track, we might say a particular variant now we believe that it causes disease, or it may be that we still don't know. So one of the really important jobs for the laboratory scientists is what we call variant curation, because when we look at a gene and find various alterations in the gene, someone has to sort out what goes on the final report and how we classify those, whether we classify them as being disease-causing or not. And keep in mind that in all of us, we have millions of variants in our genome where we might be different from the consensus sequence, or what's regarded as the standard human genome. You know, most of those would be completely benign. A handful are potentially disease-causing, and it can be very challenging to try and sort out which of those is the case. So let's talk a little bit about testing for gene variants. So the oldest type of genetic test that we have is a chromosome test. As I mentioned, this is how we used to look at chromosomes, looking at them down the microscope. And a test like this is able to detect bits of chromosome that are missing, whole chromosomes that are missing, and extra pieces of chromosome. And in our sort of pediatric patient population, where we're dealing with children mostly with neurodevelopmental disorders, you know, we find a diagnosis in about 3 or 4% of our patients doing this type of test. So it has a fairly low yield, and that reflects the fact that when we look down the microscope at chromosomes, we can only see things that are really large, that are about sort of say 5 million base pairs or nucleotides in size. So since about 2006, we have pretty much replaced looking at chromosomes with a microscope with a newer test called a chromosome microarray. And I'm not going to go into the technical details about this test, but basically it can look at chromosomes in much greater detail. And so it can see much smaller changes in chromosomes, but it's still a long way short of actually sequencing genes. So it can't see single letter changes in the genome. But nonetheless, we've made a lot of diagnoses using these tests, and in most of our patient populations, we have a yield of about 15% of patients get a diagnosis. If we want to look at individual genes or look for sequence changes in genes, typically if we know the disorder of interest, we can sequence a particular gene. And we used to do all genetic testing like this, that is one gene at a time. So if you thought that a patient had a particular disorder like cystic fibrosis, you would sequence just the cystic fibrosis gene, and you would see if you could find an abnormality. But the world, in the last few years, the world has moved very much towards testing lots of genes at the same time. And this has been a fantastic technological advancement, and it has led to many more diagnoses being made and to lots of new diagnoses. So there's different levels of test. If you're going to test for lots of genes at the same time, this can be done in different ways. One way is what's called a gene panel test, which is where you set up a test that is going to test for a defined set of disorders. And that is quite commonly done in cancer, for example, at the moment. So particularly if we're looking for a familial cancer susceptibility, we can order a test that might look for 50 or 60 genes that are linked to cancer or to another particular disorder. But in our paediatric population, and certainly in this institute, the test that we do most commonly is called an exome sequence. And when we do an exome sequence, we are basically looking at all of the known genes. So you remember the exome is about 1 to 2% of the genome. So we're focused on analysing not all of the DNA in between, but the parts of the DNA that code for genes. And so with this test, we're actually sequencing 20,000 or so genes. But at the moment, there's only 4,000 or 5,000 of those genes that have any real association with human disease. So those are the genes that we tend to focus on in our analysis. But this is the test that we perform most commonly, particularly in a paediatric population and in children with congenital abnormalities and developmental delays and so forth. And the yield of this particular test is quite high. So in that particular population, we believe that we can make a diagnosis in around half of the patients that we test. And in subsets of that population, it may be even higher. So that's a huge advance of where we were even 10 years ago. There's not really many areas of medicine where our diagnostic yield of testing or the number of patients who we can make a diagnosis has gone up that much, which is really about tenfold overall, if you consider both the advances of microarray and exome sequencing. And then the sort of, the next level up is the whole genome sequence. So that basically means you sequence everything. So not only all of the genes, but all of the DNA in between. And at the moment, this is mostly done more at a research level. And that's partly because with a lot of the stuff in between the genes, we don't actually have a good understanding about how to interpret it, what it actually means. So we can sequence it, but what you then do with the information is a little bit hard to know. So this is a powerful tool if you're trying to really discover new things. But if you want to just make a diagnosis of a known condition in a particular patient or child, probably doesn't have a big advantage over doing an exome, and at the moment it's more expensive. But I think there's a general view that within 5 years' time or so, genome sequencing will replace a lot of the testing that we're doing at the moment. So just a little bit, coming to the end, a little bit about inheritance and how genes are inherited. So the first thing to say is that when we look at gene variants, they come in a couple of different, or a few different varieties, but we talk about germline variants being variants that are present at conception. So everyone starts life as an embryo, and a germline variant means a variant was present from the time of conception. And therefore we would expect that variant to be present in all cells of the body. In contrast, Some variants are not present at conception, but arise during one of the subsequent cell divisions. So we call those somatic variants, and they may be in lots of different parts of the body, or they may just be in one part of the body. And it's somatic variants that are the main driver of cancers. So when you hear about cancer being a genetically driven condition, usually that's not something that is present at birth or at conception, but gene variants that arise during the lifetime of the patient and maybe accumulate in a particular tissue in a particular way to lead to cancer. Then we have different inheritance patterns, and I'm not going to go into detail about this, but most people are aware that genetic disorders can be inherited in different ways. They can be sometimes inherited just from one parent, so passed directly from a parent to a child. Usually we think of that as autosomal dominant. We think of that as autosomal dominant inheritance, or sometimes they can be inherited from both parents. So both parents can be carriers of a particular gene change. And if the child inherits the double dose, that is one copy from each parent, the child will have the disorder. So in this example, the father may have a gene change on one of his genes, and that may be passed into the sperm and then may end up in the child. So that's passing the condition onto the child. So when we think about types of inheritance, we have dominant, recessive, X-linked, which are conditions that primarily affect males, but can be passed on by females, and mitochondrial inheritance, which is when a condition is only passed on from the mother. And just finally, this is my last slide, but one of the kind of big discoveries of genomics over the last 10 years is just how important de novo or new gene variants are in a lot of the conditions that we see. So before genomics, we used to thought that many more disorders were inherited from a parent. Maybe they were recessively inherited. But we now realise that a lot of the conditions we see, even though they're genetic, they are not inherited from one of the parents. So they arise as a new gene change, just in either the egg or the sperm that goes on to produce the child. And often when we see families, they think that sounds, amazing and concerning, and they think that maybe they've been exposed to something in the environment that has caused this. But in fact, we know that all of us have lots of gene changes that are present in us that are not present in either of our parents, maybe, in the order of 50 to 100 in each person. And it's really just by good luck in most of us that one of those gene changes doesn't happen to land on a gene that's particularly important. And it's been estimated that maybe about 1 in 300 of all babies that are born have a genetic disorder caused by a de novo gene change. So it's quite an amazing number, and makes that a very large contributing factor to a lot of the conditions that we see. I guess the key message there is that often we end up saying to patients that just because something's genetic does not mean it's inherited, because people often treat those two terms somewhat synonymously. The good news about finding that a lot of conditions are due to de novo or new gene changes is that we can be much more reassuring to parents if they want to have another child, that the risk of having another child with a particular disorder is very low. So I got to my last slide in 23 minutes, so that was not bad time management. Does anyone have any questions?
Hi, David. Thank you for your presentation. I just had a question about exome testing. And you talked about it as identifying particular variations. How important is it to, I guess, identify what you're focusing on for example, when you do an exome and you say you're looking for things that might explain intellectual disability, what happens if there's something else? Is that found through an exome, or do you need to specify it at the beginning when you're starting the search through an exome?
Yeah, that's a really good question. So when with exome sequencing, obviously you end up with vast amounts of data, and somehow that has to be sorted through to tease out the things that matter, or maybe the one thing that matters, which could be the diagnosis, and throw out the stuff that is just background noise that is not important. And as in any sort of filtering process, there's a risk that you get it wrong if you set the filter at the wrong level, okay? If you're too stringent and say you only want stuff that is blaringly obvious, you may well miss the cause of a child's problem. On the other hand, if you let too much in, you can be overwhelmed with like thousands of bits of information and, you know, which of these is important. So I think this is part of the process of sorting through the exome. And, you know, a critical piece of information is the clinical stuff about the child or the adult, if it's an adult being tested. So we do like to get as much clinical information as we can to help guide the interpretation. So if you were in one of the reporting meetings, someone might say, well, we found this in a particular child, and the clinician might say, well, this child's nothing like what we would expect for that condition. So we would downgrade that based on the clinical information. I guess if we're talking about intellectual disability, one of the difficulties is there are so many intellectual disability genes. You know, there is over 1,000, and that's a pretty overwhelming number to start with. And a lot of the newer diagnoses that we come across are quite non-specific. So the kids might have intellectual impairment and maybe a couple of subtle features, but not much else to guide us. So I guess to cut a long story short, it's complicated and we use a lot of information, but we may get it wrong occasionally. Yeah.
I'm interested to know your prediction moving forward regarding affordability and accessibility of exome testing, and how does Australia rank compared with other countries in the world regarding accessibility for our children and young adults who perhaps missed out on diagnosis because of the changes that we now have. I work in speech pathology and I have a lot of clients presenting who could benefit from exome testing, but perhaps families aren't aware, or perhaps they see it as being primarily unaffordable and inaccessible.
Yeah, so the question is about affordability and accessibility of genomic testing. And I guess we still have a long way to go, I would acknowledge. I would say, however, we have made a vast leap in the last 3 or 4 years. So I think 5 years ago, I would have said Australia has really fallen behind a lot of the rest of the world, particularly Europe and the US, in terms of the availability and accessibility of this type of testing, because we weren't really doing it at all for these pediatric populations. Now we are doing a lot. We are testing, you know, dozens of patients every week, but there is still a fair way to go in terms of accessibility. I think we will get there in the next few years. I would say, you know, in 3 or 4 years' time, my expectation is that, you know, every child who fits into one of these diagnostic groups would have access, whether that's through the hospital system or through Medicare funding.
So that just leads me to ask the inevitable question regarding NDIS. And I'm sorry to bring that up so early in the day for everyone. Obviously, we've got a process of filtering clients in through NDIS? What's the role of genetic testing in relation to accessibility of NDIS funding based on syndromes? And I'll just couch that in one example of a client who had a known syndrome. In order to support this client, it took me 12 months to actually work through the local member, go on Facebook and find other individuals who had the same syndrome in Australia who'd been funded for early intervention through NDIS, and yet one individual within the system had deemed that this person was ineligible. And so it took 12 months to actually get her funding in spite of her diagnosis of a syndrome.
Yeah, so I don't claim to be an expert on the NDIS. However, in principle, the NDIS is not supposed to be diagnosis-based. It's supposed to be disability-based. And, you know, I am a strong supporter of disability-based support rather than diagnosis-based. You know, in the previous system, there was diagnosis-based access to funding, and clearly that disadvantaged people with ultra rare disorders whose condition was never going to make it onto a certain list because it just wasn't common enough. With a disability-based system, you might expect some children with a given diagnosis to have access to funding and others not to, because every child with a given diagnosis is not the same. We know there are conditions where some children have a significant disability, others don't with the same condition. Having said that, our experience is that having a diagnosis for some families seems to help them get noticed or by the NDIS system. So we do not uncommonly provide letters of support, for example, to say this child has a given diagnosis and, you know, this is what might be expected or and so forth. So I'm not sure whether that answers your question. I would say it shouldn't be, the access to funding shouldn't be reliant on a diagnosis. It does seem to help a little bit. And I guess we understand that, and that's another motivator for us to try and get diagnoses for patients.
Hi, Chris. I'm just going to, if you don't mind, David, jump in a bit. We were in Canberra on Thursday for the rare disease function up there. Yeah, Rare Voices. And that came up with Bill Shorten, and he heard loud and clear that for sure that rare disease patients are struggling to get access, and that it's something that he wants to fix. Having said that, if it's a known syndrome and there are other families around, or there is a support group, then I would contact the support group and ask them to write you a letter saying this is the kind of presentation that they, you know, commonly in that gene they have. Yeah.
Can I just answer that? It is tricky with NDIS. It's meant to be a diagnosis, a needs-based scheme, not a diagnosis. Just put your dot points down, how it affects them with a disability, and I can talk to you more about it. It is an issue. We are lobbying about it. We did mention it in our planning submission, and it'll be mentioned in the Act review as well for rare conditions.
I actually have a question as well. With my hat on, I'm the parent of a child with SCN2A, which is a rare genetic condition. And we have families that get the VUS variant of uncertain significance. What would be the precedent for that to change to a pathogenic? Because these kids are presenting with very similar symptoms, very simple presentation, you know, feeding issues, epilepsy, etc. But they're given a variant of unknown significance, which puts them in this, well, has it caused my child or hasn't it? Like,
Yeah.
what, yeah, so how does it change, and what would you suggest for a family that's got that?
Yeah, so just to make it clear that the classification, the VUS classification, is a laboratory classification, and it's based on how they assess the evidence at the time, okay? In terms of what goes back to the patient, that needs to be combined with the clinical assessment, that is the geneticist or the doctor who is seeing them. So we might see a patient and say, well, the report says VUS, but we are pretty certain that this is the cause of your child's problem, and we would consider that's the diagnosis. And in the fullness of time, more evidence will come. Maybe another child in the world will be found to have the same variant and the same phenotype, and that will be enough to push this towards a pathogenic mutation. Conversely, the geneticist might look at the report and say, well, it's a VUS, but it doesn't match the child at all. So I think we can disregard it. But you can't just take the lab report and put the clinical interpretation based on the lab. It needs the geneticist's opinion. And hopefully for these people you're talking to, I would say, go back to your geneticist and say, what do you think?
Not close enough. Hello, we're parents of a child with an extremely rare genetic condition. He was diagnosed about 3 years ago, and his karyotype's the only type in the world. So we're fortunate to have a diagnosis, but we're in the position that the diagnosis doesn't necessarily mean a lot because it's so rare. A geneticist interested in the medical presentation and possible implications of completely unique chromosomal deletions? Is that something, like, I guess I'm wondering, he's 5, so we're obviously hoping in his lifetime that we'll gather more information, but from a geneticist's point of view, is that a priority or a possibility?
Yeah, so chromosome disorders are a special category, partly because the vast majority are actually unique. So there's a handful of chromosome disorders, I guess Down syndrome being the very well recognised one, where it's the same genetic change we might see in dozens or hundreds of children, but they're the exception rather than the rule. So most parents who have a child with a chromosome problem, the chromosome problem is unique to that child, because when you think about it, to have a chromosome deletion, you need 2 breakpoints, which are often randomly distributed across all those billions of nucleotides. What's the chance you'll have 2 anywhere in the world that are identical? Pretty low, okay? So from a research perspective or a geneticist perspective, sometimes we try and, if someone has a particular interest in a group of disorders, yes, we try and collect data and we try and compare deletions that are similar, and we might put them out in a publication. Occasionally, we might just publish a child who we think is unique and see what comes back. Okay, keep in mind that there's a lot of kids in the world that are not published. So where we have parents who are really motivated to say, we want to find out more about what's going on with our child, sometimes we might just say, well, let's write it up and put it in the literature and see what comes back. And it might be nothing, or sometimes other people come out of the woodwork and write to us and say, we've got a child that's just the same, let's compare notes. And I guess I'd also say, we do have international databases that we can kind of enter our patients into, and that's quite useful as well. So once you enter the patient in that setting, I don't know whether your child is, but then sometimes we get, emails out of the blue from people elsewhere in the world, and sometimes the parents just want to be put in touch with each other and to compare notes, and sometimes they're interested in research. Yeah.